Welcome to the deep dive. Today we're zeroing in on polydimethylsiloxane, PDMS.
Right, PDMS. It's this silicone-based polymer that honestly pops up just about everywhere. You see it in cell biology research, lots of biomedical devices.
It's pretty ubiquitous.
Exactly. And for you, our listener, the learner, getting a handle on PDMS is, well, it's more than just academic. It's elasticity, the fact that you could basically tune how stiff or soft it is. That's fundamental.
How so?
Well, think about growing cells on a substrate.
Yeah.
How stiff that substrate is really affects how cells behave. And for microfluidics, those tiny lab-on-a-chip devices, its properties are crucial. So yeah, if you're into biomaterials or bioengineering, this stuff matters.
Okay, so to really dig into this, we've got 3 research articles. Each one looks at the mechanical side of PDMS, but using different methods.
Yeah, different scales too. We've got nanoindentation, that's super tiny probing.
Right.
And then macroscale tests, like compressing it and stretching it, which are maybe more familiar concepts.
So our mission today, we want to pull out the main ideas on how PDMS elasticity gets measured, at these different scales, and really focus on how that cross-linking thing works, how changing the recipe changes the stiffness.
And we'll definitely keep an eye out for where the different methods maybe don't quite agree.
Yeah.
Because sometimes those little discrepancies, they actually tell you a lot.
Good point. So where do we start? What's the absolute key to its elasticity?
Cross-linking, hands down. That's the first big takeaway from all the sources. It's all about the connections, the cross-links within the polymer network itself.
Okay, crosslinks, like little bridges holding the polymer chains together.
That's a good way to think about it. And the more bridges you build, the more rigid the whole structure becomes. So by controlling the density of these crosslinks,
You can basically dial in the stiffness you want, maybe make it feel like soft tissue or something a bit firmer.
precisely, which is why it's so great for those cell growth studies,
Yeah.
trying to mimic different body tissues. And the main handle we have on controlling this is the mix ratio.
The ratio of the base polymer to the curing agent, right? I saw numbers like 5:1, or even way out at 33:1.
Exactly. More curing agent means more crosslinks, which means stiffer PDMS. Simple as that, in principle.
And one paper even gave a kind of rule of thumb.
Yeah, it was interesting. They suggested that the elastic modulus, that's our measure of stiffness, in megapascals or MPa, is roughly 20 divided by that base to curing agent ratio.
So like 20 divided by 10 for a 10:1 mix gives you around 2 MPa.
Kind of, yeah. Now, you got to remember that's a generalization from one study's conditions, but it gives you a feel for the trend. A higher ratio number means softer material.
Got it. Okay, so if cross-linking is the key, how do they actually measure the resulting stiffness? You mentioned macroscale compression first.
Right. One group actually built their own compression testing device. It sounded quite custom.
Like a bit of a science project.
Maybe a very sophisticated one. They use a heavy granite slab for stability, a vertical post, a digital depth gauge, but they took the spring out for better sensitivity.
Ah, clever.
A platform for weights, and a flat cylinder, a punch to press down on the PDMS samples. Pretty neat, actually.
So the idea is just put a weight on, measure how much it squishes down.
Fundamentally, yes, but they did mention doing a preload.
Why do that? Just put the weight on, surely.
Well, the preload is important to make sure that flat punch is sitting perfectly flat and in full contact with the PDMS surface right from the start. If you don't, you might get this initial kind of squishy bit in your data, they called it a toe effect, that isn't really the true stiffness.
Oh, okay. Avoids artifacts from uneven contact. Makes sense. So what stiffness values did this compression setup give them?
They found it varied quite a bit with the ratio, as you'd expect. It went from about 0.57 MPa, that's quite soft for the 33:1 mix,
Okay.
all the way up to 3.7 MPa for the much stiffer 5:1 mix.
Wow, that's a big range, about 6 or 7 times stiffer.
Exactly. And importantly, they saw a nice, clean, linear relationship. More crosslinker meant proportionally higher stiffness, at least within the range they tested. And yeah, their results supported that E is roughly 20 divided by the ratio rule of thumb we mentioned.
Okay, so that's compression. But another study looked at stretching at macroscale tension. Different approach entirely.
Yep, tensile testing. They used a standard procedure, prepared specific sample shapes, and pulled them, measuring the force and how much they stretched. The interesting part is comparing these results to the compression ones.
Right. Did they find the same stiffness values?
Well, yes and no. They also saw stiffness increase significantly as they added more crosslinker. It went from 0.018 MPa, which is really, really soft.
Almost jelly-like.
Pretty much, yeah. At about 2% crosslinker, up to around 1.5 MPa at 9% crosslinker. So the trend is the same. More crosslinker, more stiffness.
I sense a but.
The but is, especially for those softer samples, the ones with fewer crosslinks, the stiffness they measured when stretching was much lower than what the other group found when compressing similar materials.
Lower in tension than compression. Why would that be?
It suggests that for these less cross-linked networks under tension, the material might behave, well, almost more like a very thick liquid than a true solid. The chains might be rearranging or flowing more easily when pulled apart compared to when pushed together.
Huh. That's counterintuitive almost.
It is, and it shows how complex PDMS can be. To try and model this mathematically, they use a specific type of S-shaped curve, a sigmoid function, technically the Boltzmann equation, to relate stiffness to the crosslinker percentage.
Okay.
But crucially, the exact shape of that curve, the parameters they plugged into the equation, were different for the compression data versus the tension data. It really reinforces that the material responds differently depending on how you deform it.
So compressing and stretching tell slightly different stories, especially for softer PDMS. Now, what about going really small? Nanoindentation.
All right, let's zoom way down. Nanoindentation basically uses a tiny sharp tip to poke the material's surface and measures the force and depth very, very precisely. It gives you localized mechanical properties, great for soft stuff like PDMS.
But you said there are challenges. It's not just poking it.
Not quite. The main headache with PDMS at this scale is that it's sticky, tacky.
KT, how does that interfere?
It makes it really hard to tell the exact moment the tip first touches the surface. There are these tiny adhesion forces that can sort of pull the tip in before you even apply much load. And knowing that exact contact point and the contact area is critical for calculating stiffness accurately.
Okay, so how do they deal with the stickiness?
Clever ways. Sometimes they do the tests inside an electron microscope so they can literally see the tip make contact. Or they use dynamic methods where they oscillate the tip slightly, which can help separate out the different effects.
And they use different shaped tips too. I remember seeing Berkovich, flat punch, and spherical ones mentioned.
Exactly. One study directly compared a Berkovich tip, it's a standard three-sided pyramid, with a flat cylindrical punch tip in what they call quasi-static tests, just pushing in slowly.
And did the tip shape matter much?
Interestingly, once they accounted for that initial adhesive pull-in with the sharp Berkovich tip and made sure the flat punch was fully in contact, they got pretty similar results for a 5-to-1 PDMS, around 1.5, 1.6 MPa.
That's reassuring. Similar to the Macroscale tension result for stiffer PDMS, actually.
It is, yeah. But they also noticed something else interesting in those tests. When they pulled the tip out, the unloading part of the curve, the material seemed stiffer than when they were pushing in.
Stiffer pulling out than pushing in. Why?
That's the viscoelasticity of PDMS showing up. Yeah. It's not perfectly elastic like a spring. It has some viscous or fluid-like damping behavior too. Think honey versus a rubber band.
Okay.
So some energy is lost during the deformation, and the response depends on how fast you deform it. The higher stiffness during unloading suggests some of that deformation isn't recovered instantly. It takes time.
Right, that time-dependent aspect. And what about the dynamic nanoindentation? You said that gave higher values.
Yes, nano-DMA, or dynamic mechanical analysis at the nanoscale. They oscillate the tip at specific frequencies. And they found the storage modulus, which measures the elastic part of the response, increased as they tested at higher frequencies. For that same 5-to-1 PDMS, it reached around 3.5 or 3.6 MPa.
Wait, 3.6 MPa? That sounds familiar.
It does, doesn't it? It's actually very close to the value the other group got from their macroscale compression tests on 5-to-1 PDMS, which was 3.7 MPa.
Whoa. Okay, so dynamic nanoindentation matches macro compression better than static nanoindentation or macro tension, at least for this stiffer sample.
It seems that way, yeah. It suggests that the rate of deformation plays a big role, and maybe the fast oscillations in nano-DMA mimic the conditions in macro compression better in some respects. It definitely highlights how testing conditions, static, dynamic, frequency, depth, all influence the number you get.
Man, it really isn't just one number for PDMS stiffness, is it? It's a whole landscape.
You got it. And then there's another nano-level technique designed specifically to handle that stickiness issue, the nano-JKR method.
JKR, what's that about?
It uses a spherical indenter tip and directly accounts for the adhesion forces between the tip and the surface. It's based on the Johnson-Kendall-Roberts theory of adhesive elastic contact.
So it embraces the stickiness instead of just trying to avoid it?
Kind of, yeah. They carefully measure the full force curve, pushing in, pulling out, and importantly, capturing the pull-off force needed to finally break the adhesive contact. By analyzing that curve, especially the unloading part and the pull-off details, they can calculate the elastic modulus while accounting for adhesion.
Clever. And what stiffness values did nano-JKR give? Different again.
Somewhat, yeah. The nano-JKR values tended to be lower than macroscale compression results. For instance, for a 10-to-1 ratio, they got about 1.24 MPa, and for a very soft 50-to-1, it was only 0.1 MPa.
1.24 MPa for 10.1. That's maybe closer to the tension results for stiffer samples rather than compression.
That's a good observation. It seems nano-JKR might align a bit better with tension, or perhaps represents a different aspect of the material response compared to the bulk compression. They could also fit that same sigmoid curve shape to the nano-JKR data versus crosslinking, just with different specific parameters again.
Okay, so JKR is good for sticky samples with spherical tips, but I saw they even tried to apply similar ideas to the sharp tips, like Berkovich, using computer simulations.
That's right, pretty advanced stuff. They used numerical modeling to simulate the contact, incorporating adhesion using things like the Lennard-Jones potential, and calculating surface deformation. It was an attempt to extend the JKR-like analysis beyond just spheres.
And did the simulations match the experiments?
So the 5-to-1 PDMS, the simulations using sharp tips like a cube corner and a Berkovich, actually gave modulus values around 3.6 to 3.8 MPa that agreed surprisingly well with the macroscale compression results again.
Interesting. So the simulations bring the sharp tip nanoindentation back in line with bulk compression.
In this case, it seemed to. A conical tip simulation gave a slightly lower value, around 3.4 MPa, but it suggests that if you can properly model the complex tip geometry and adhesion, maybe the underlying stiffness being probed is closer to the bulk compressive modulus.
But simulations are models, right? They mention some potential reasons for lingering differences.
Oh, sure. They noted things like, the real indenture tips aren't perfectly sharp or perfectly shaped like the models assume, and the mathematical approximations used for adhesion might not be perfect for sharp tips. Plus, the models often assume small strains and purely linear elastic behavior, which might not be fully accurate for PDMS under indentation.
Okay, wow. That's a lot of different ways to poke and prod PDMS, so let's try and bring it all together. Compression, tension, nanoindentation with various tips and methods.
Yep.
What's the big picture?
The big picture is, it depends. Seriously, how you measure PDMS stiffness really matters. Macroscale compression seems pretty reliable, especially for the stiffer formulations. Nano-JKR, with its focus on adhesion, seems better suited for the softer, stickier end of the spectrum. We consistently see PDMS as less stiff in tension than compression, particularly when it's less cross-linked, that almost fluid-like behavior idea.
Right.
And the dynamic nanoindentation, the nano-DMA, can actually give results quite close to macro compression, suggesting that the rate of testing is a key factor connecting micro and macro scales.
So summarizing this deep dive then, the key takeaway is PDMS stiffness is super tunable through crosslinking. That's job number one.
Absolutely.
And while different measurement methods give different numbers, sometimes quite different, they often show similar trends, and the differences themselves tell us something about scale, surface effects, or viscoelasticity. That sigmoid curve relating crosslinking to stiffness seems like a useful recurring theme too.
Definitely. And hopefully for you, the learner, this has given a pretty comprehensive overview without getting totally lost in the weeds. A shortcut maybe to understanding how we characterize this really important material.
Absolutely. So let's leave everyone with a final thought. We've seen these discrepancies, right? Between tension and compression, between static and dynamic, micro and macro. What fundamental things about PDMS might we still be missing? And how could these subtle differences in measured properties actually impact how PDMS performs in, say, a flexible sensor versus a cell culture? Are we measuring the right thing for each application? And maybe, just maybe, do we need entirely new ways to probe materials like this to get the full picture?
That's the million-dollar question, isn't it? What else is hiding in the behavior of this seemingly simple polymer? Lots to think about there.